Okay, so saya nak refresh again the process of DNA replications. Okay, DNA replications is the process that occur during S phase. Okay, S phase during interphase. Okay, interphase during cell cycle. Okay, for in the topics of cell divisions. Okay, for the DNA replication, as I mentioned before, okay, we have to list down first enzyme and protein involved in the process of DNA replication. First is DNA helicase. Okay, functions of DNA helicase to unwind DNA double helix. Okay, number 2, single-stranded binding protein. Okay, single-stranded binding protein untuk stabilize strands of DNA. Actually, single strands of DNA. Okay, number 3 is topoisomerase. Okay, untuk release tensions or supercoil. Number 4 refer to the primase to catalyze the additions of RNA nucleotide. And number 5, DNA polymerase. Okay, they have 1 and 3. 3 for additions of DNA nucleotide and proof reading. Okay, once untuk replace, actually remove and replace. Remove and replace. Okay, RNA primer. And number 6 is DNA ligase to join Okazaki fragment. Okay, so remember, number 1 DNA helicase, number 2 single-stranded binding protein, number 3 topoisomerase, number 4 primase, number 5 DNA polymerase, and number 6 is DNA ligase. Okay, starting with we look at the process of DNA replication. Okay, so ingat number 1 until number 6. Okay, first this is a DNA strand. Okay, DNA strands with the nucleotide base. Okay, and then number 1 DNA helicase bind to the origins of the DNA replications. Origin means starting point for the DNA replications to occur. Okay, and we know that number 1's DNA helicase ni function to unwind the DNA double helix. Okay, so the double strands of the DNA will become a single strands of DNA. Okay, so this is DNA helicase. Okay, and the single strands of the DNA, after it's open the DNA double strand, the single strand is become unstable, and it will be stabilized by number number 2, single-stranded binding protein. Okay, to prevent the single strands of the DNA to reforms. Okay, ataupun to reforms the double helix. Okay, at the ends of the replication fork, this is we call a replication fork. Okay, they will form supercoiling. Okay, because kita paksa dia open. Okay, this supercoiling ni will be released by topoisomerase. Okay, and the strands become be like this. Okay, without supercoiling. Number 1, number 2, and then number 3. Okay, and then next is number 4 is primase. Okay, primase akan datang sini, akan come here. Okay, why here and here? Okay, due to the rules of the DNA replication. Okay, DNA replication will synthesis new strands of DNA in 5' to 3' directions and use template of DNA 3' to 5' of DNA. So remember back, the structures of the DNA is anti-parallel. Anti-parallels means they have huruf pula. Okay, anti-parallels means we have 5', 3'. So 3', 5'. Okay, 5' refer to the NOH presenting at the 5 end, and another part is presenting at the 3 end. So refresh back the structures of the DNA in chapter 1. Okay, and because of the rules is use template 3', 5', so here is 3'. Okay, and here is 5'. So here is 5' due of the natures of the anti-parallel. 3' here. Okay, maknanya the template is starting from here and this starting from here. Okay, RNA polymerase will attach at the 3 ends of the DNA template. Okay, and they will add the catalyze the additions of the RNA primer here. Okay, and then followed by DNA polymerase 3, number 5, DNA polymerase 3. Okay, number 5, DNA polymerase 3. Okay, DNA polymerase 3. Okay, we'll add DNA nucleotide. Okay, here and here. Okay, follow the rules of synthesis new strands of DNA from 5' to 3' directions. Okay. Okay, why the movements first move towards the replication fork? Ini kita dah mention the replication fork, and the another is away the replication fork due to the natures of the DNA is anti-parallel. Okay, so imagine the strands of the DNA ni open more. Okay, when it's open more, okay, this RNA polymerase tadi. Okay, this RNA polymerase and DNA polymerase will continue additions of the DNA nucleotide and elongate towards the replication fork. Okay, and then this one, so this Okay, one is open up. So RNA primase will add the new RNA primer, because this starting with the 3 prime. This is 5 prime. Okay, follow this. Kita ada 5 prime here and this 3 prime. Okay, so 3 prime here. So they will add another new RNA primer, and the DNA polymerase 3 will add DNA nucleotide. Okay, away the replication fork. Okay, and if we continue, okay, for example like this. So this will continuously elongate towards the replication fork, while the other one will form discontinuous of the discontinuous of the strands that away from the replication fork. Okay, ni continuous, this one is discontinuous. Okay, this short fragment we call as Okazaki fragment. Okay, this strand we call as leading strand, discontinuous strands, and these strands that away from the replication fork we call as lagging strand. Okay, we need to form DNA. So the DNA polymerase 1, okay, number 5, DNA polymerase 1, okay, will remove. Okay, it's remove. Okay, it's remove RNA primer. Okay, remove RNA primer and replace it with the DNA nucleotide. And finally, they will form structure like this. Okay. Oh, wait, sorry. Okay, and they will form structure. Sorry. And then they add DNA nucleotide, be like this. And then number 6, this Okazaki fragment will be 6 will be joined by the DNA ligase, and finally, okay, they will produce 2 identical strands of the DNA that consist of old and new strands. Okay, 2 identicals of DNA, and each strand consists of old and new strand. Follow semi-conservative model.